Hi there, my name is Lawrence Hunt, and this video is going to tell you about our recent paper that we've just had published in Nature Neuroscience, titled Triple Dissociation of Attention and Decision Computations Across Prefrontal Cortex. In this video, I'm going to be telling you about the neurophysiological recordings that we made in 3 different subregions of macaque prefrontal cortex. And these recordings were being made whilst the subjects were performing an attention-guided decision-making task in which they were trained to saccade to different cues in order to gather pieces of information that would lead to them making a reward-based choice. What we found was that by using a representational similarity analysis of these neural recordings, we could very clearly dissociate what features of a task were being represented within each of those 3 different subregions of prefrontal cortex. And this happened right from the time when the very first piece of information relevant to the decision was being attended by the subject, and that's what's being shown in this video here. And we could also investigate how the neural signals evolved across time as more cues were sampled by the animals, leading right up to the time when the animals made their final choice by moving a joystick. And what we found was that orbitofrontal cortex, OFC, carried information reflecting an attention-guided value comparison process, but by contrast, anterior cingulate cortex, ACC, carried several signals that evolved across time, leading to a final commitment to a choice. And that's what's shown in this video here. So these signals included belief updating in light of newly attended information, and also an emerging plan of what action the animal was going to choose in order to make the decision. So if you'd like to understand a bit more about how these analyses were performed in the paper, what we discovered, and what we think it means for understanding decision-making, then stay tuned to this video and I'll try and provide a full explanation of our results. Okay, so before I begin, I just want to mention a few things. First of all, although I'm now based at the Wellcome Centre for Integrative Neuroimaging at the University of Oxford, as you can see at the bottom of the screen here, this work was actually conducted during my postdoc at University College London in the lab of Steve Kennerley. And it was very much a joint project between myself and a PhD student in the lab, Malalasekera, who's the joint first author on the project. And Nish really did the lion's share of the data collection for the project, and I did the majority of the data analysis. And so it really was a team effort between us, along with the input from the other authors who are listed here on the paper. The second thing to mention is that the paper is available at this DOI here, but you can also freely download the raw data for the paper because we've uploaded it to the CRCNS website, a collaborative resource for computational neuroscience. And that repository includes all of the MATLAB scripts to reproduce all of the figures from the paper. And we'd be really excited to hear from you if you've downloaded the data, especially if you've tried out any new analyses on the data set. The third thing to mention is kind of what I'm trying to do in this video. And I guess the aim of this video is to maybe provide a bit more background to our analyses, and also maybe some of the philosophy behind where the experiment came from, and go into that in a little bit more depth than we had to in certain places in the paper. So I'm hoping that the talk should be accessible to people who've come from slightly different fields and slightly different backgrounds, but especially to graduate students, to new PhD students and master's students, maybe even advanced undergraduates who are new to cognitive neuroscience. And so in order to try and make the talk maybe a little bit more interactive, what I might try to do in a couple of places is probably just to try and pose a question to you, the listener. And if you're a student and you want to, you can just pause the video at that point and see if you can figure out what you think the answer would be before continuing with the next section of a video. So I hope that doesn't seem too clunky, and hopefully it's useful and interesting if you're encountering this material for the first time. And then the last thing I want to mention is that a few of the figures that you'll see in the talk are slightly different from how they appear in the paper. Either the colour scheme may have changed, or in a couple of places even the data might appear slightly different from how it's presented in the paper. The reason for this is simply that this talk was actually originally written before we'd submitted the manuscript. And then, of course, we had to make a bunch of edits to the manuscript and a few changes to some of our analyses in response to comments from peer reviewers and also to comply with formatting from the journal. But all of the key results still hold exactly as in the paper. And what I've also tried to do is to make sure that the figure number from the paper always appears normally in the bottom right-hand corner of the screen. So you can always refer back to the paper to see the final peer-reviewed version if you want to. Okay, so with all of that preamble out of the way, let's begin the talk. So this project was designed to look at reward-guided decision-making in primates. And here are some primates out making reward-guided decisions. This is, of course, the famous photograph, 99 Cent, by the photographer Andreas Gursky. And I like to think that one of the many reasons that this picture has become so famous over the years is partly because it kind of sets into contrast the almost bewildering array of choices that we might be faced with, even on like an everyday visit to the supermarket, versus the kind of facile, almost boring nature with which we go about solving these choices. I mean, this is not a difficult decision problem for us to solve, even though we have an inordinate number of choice options that we might be comparing. And the reason why we can make decisions such as this one is, of course, that we don't consider and evaluate all of these different alternatives together, but instead we treat the decision as a sequential problem. So what we do is we consider one option at a time, and we go around and we deploy attention to each alternative. And so the question that we wanted to ask in this paper is basically how we go about that process of selecting between alternatives under the guidance of visual attention, and how our shifts in attention might shape some of the computations that are performed in prefrontal cortex when we make those simple value-based choices. And I think that this is not only important because it underlies decision-making of the kind that we see here, but also, of course, because how we choose what course of action is going to be rewarding is essential to almost all of our daily behaviour. Okay, so how have people tried to study decision processes like this in the lab? Well, what we might do is we might take somebody and we might put them inside a scanner, so an fMRI scanner, as you can see here. And we might ask them to perform a simple task. And the kind of task we might ask them to do would be where, for instance, two food items might flash up next to one another on the screen. And the subject would be instructed to select which one of those two food items he'd most like to eat with a button press. And this is, of course, what we'd think of as an example of two alternative forced choice. What's nice about this paradigm is that it kind of reduces some of that complexity of what we've seen in the naturalistic decision scenario to a relatively simple thing that we can study in the lab, where we can place everything under very tight experimental control, so we can control what stimuli are being performed, and there are only two items to choose between. And we can also take models that we know describe the value of those different items, for instance from economics, and use those as regressors to explain activity that we see in the brain whilst those choices are being made. And so this, of course, has given rise to this field that's been referred to as neuroeconomics, where the decision theory borrowed from economics is used to explain neural activity. And what's appealing is that it's allowed us to try to build up some kind of toy models of how these decisions might unfold. So here's one example of this from a review paper by Camillo Pardo-Eschiopera, now a few years old, but still highly influential on the field. And what it proposes is the way that people solve these kinds of decisions, where you have to maybe compare an apple and a chocolate bar, is that you first have to extract some features of what those stimuli actually consist of, and that might take place in sensory areas and limbic areas. And then you might have to map those different features into some kind of unitary space of value. And value would, in this framework, be represented in places like orbitofrontal cortex, OFC, and ventromedial prefrontal cortex, VMPFC. And the idea would be that by taking these two things, which are really quite different from one another, one's an apple and one's a chocolate bar, and mapping them into this common space of how valuable they are, you can then compare them with some kind of common currency. So you can basically say, how much do I prefer eating a chocolate bar relative to eating an apple, for instance? And so by having explicit representations of those values, we can then compare the values of the different items and turn that into an action plan for what action we might want to make in order to commit to making a choice. And that might take place in regions such as lateral prefrontal cortex or anterior cingulate cortex. And then finally we can translate that into the movement, the button press, executed by regions in the motor cortices and the basal ganglia that would allow us to choose which one of those two buttons to press and which choice we want to make. Okay, so that's a kind of toy model of how those decisions might be solved in the brain. But if we think back to the example in the supermarket, I think that we realise that there also must be some other factors that influence the way in which we make choices for rewards that require us to think about some other processes beyond what are explained in those simple neuroeconomic models of binary choice. And the key thing that I want to emphasise about decision-making today, both in humans and in other primates, are that most decisions that we make are inherently sequential in nature, and they're heavily shaped by visual attention. And you can see this here. So this person shopping in the supermarket might look at each of the different items as he deliberates, weigh up the costs and benefits associated with each of the different items. And there might also be examples, just like the second one in that video, where he just considered one of the items on offer, and he decided that he liked it so much that it didn't even seem worth considering any of the other possible choice alternatives. So he only really attended to one of the items and committed to choosing that item. So that immediately gives rise to some questions. How does this person making the decision decide whereabouts he's going to look next, whereabouts he's going to deploy his attention as he makes the choice? How does his shifts in gaze between the different items affect the eventual decision that he made? Whenabouts did he decide that he'd sampled enough information and could commit to making a final choice? And most importantly, from the point of view of this talk, how do all of those processes of redeployments of attention shape the computations that we think are taking place in different subregions of prefrontal cortex as that decision process is unfolding? I think that a second reason why we might start to think about decision making from this more sequential perspective, particularly in primates, is that these might be the kinds of decision that primates have evolved to make. So in some ways, the sequential decision that I just showed you in the supermarket is kind of similar to the kinds of decisions that other primates make when they're out foraging for food. So here are two monkeys who are foraging in very different environments. One in the snow in Japan, and one is a squirrel monkey. But there are some features of their behaviour that are common. They both rely very heavily upon visual attention as they search for food, and they both perform that search in a very sequential fashion. In a really fantastic book on the prefrontal cortex by Dick Passingham and Steve Wise, you can see the reference at the bottom of the screen there, what they've argued is that the primates forage in very distinct evolutionary niches from other mammals. So in particular, they're diurnal rather than nocturnal, and their foraging relies heavily upon visual attention. They have forward-facing eyes and a fovea, which allows really fine visual discrimination. And they also have dexterous hand-eye coordination, which means that they gather information about what to do with their eyes, and then they collect those rewards with their hands. And they can also learn extremely rapidly, based upon visual cues, about what features of their environment are likely to lead to reward, and which items are not. And they often rely just upon individual visual cues in order to shape what course of action they're going to take next. And what Passingham and Wise then go on to argue is that this might be the reason why, at the same time as primates had a very dramatic expansion of the visual system and the development of fine hand movements, they also showed this very dramatic expansion of prefrontal cortex. And in particular, they developed a granular prefrontal cortex with an expanded layer 4, which of course has then become further expanded in humans, but is entirely absent from other species, so for instance from the rodent. So I think that if we're going to understand how different subregions of prefrontal cortex support reward-based decision-making, it makes sense to try to study it in primates, but it also makes sense to try to understand the role that visual attention plays in shaping our choices. Now, we're certainly not the first researchers who have been arguing along these lines, and there have been a number of really pioneering studies that have begun to address some of these questions, both in the domain of economic decision-making and also in the domain of foraging decisions, particularly using human functional imaging. So in particular, work from Antonio Rangel's group in Caltech has argued that economic decision-making can be really well described using a sequential sampling decision model, such as what's known as a drift diffusion model of a decision process. But what's important is that they showed in this paper here by Ian Krybeck that these processes can be substantially improved in terms of how well they describe economic choice if we consider the role that attention plays in shaping the decision process. And work from the same group has also then shown that the bold fMRI signal that you measure in ventromedial prefrontal cortex seems to reflect the relative value of the item that you're currently attending to minus the value of the item that you previously attended to when you're forced to shift your attention back and forwards between different items. And that's what's shown in this paper in Neuroscience in 2011. On the other hand, studies of foraging style decisions have maybe tended to focus on a slightly different structure, which is the anterior cingulate cortex. Single unit recordings made by Ben Hayden in Michael Platt's lab showed that in those kind of patch leaving decisions that I just showed you made by macaque monkeys, then there are neurons within the anterior cingulate cortex that ramp up towards a decision threshold at the very point at which the animal decides to leave the current patch and go and forage in a new location. A similar account has also been used to explain the bold fMRI signal, again within the anterior cingulate cortex, which has been argued to reflect the relative value of foraging versus searching. That was work done by Niels Colling and Matthew Rushworth in Oxford. But what I think these studies haven't yet told us is actually what's really being computed or represented within each of these different subregions of prefrontal cortex as the decision unfolds. And also how these representations might evolve across time in order to support a reward-based decision guided by visual attention. So what we set out to try to do in this study was to make some recordings both in the orbital and ventral regions, which are emphasised kind of on the left-hand side of this slide, and also from the ACC region, which is emphasised on the right-hand side of the slide, and also from a third region, dorsolateral prefrontal cortex, whilst monkeys were making a reward-based choice based upon visual attention. So what we did in the study was to record from 2 macaque monkeys, and we positioned our recording chambers so that we could have access to all 3 of the regions that are shown on this slide. So the dorsal bank of the anterior cingulate sulcus, which is shown in red, area 13 of the orbitofrontal cortex, which is shown in blue, and the principal sulcus in the dorsolateral prefrontal cortex, which is shown in orange. And what we could do was lower multiple electrodes acutely into each of these regions. And with our recording setup, we could typically record somewhere between 10 to 40 neurons a day from these regions. So we could get a reasonably good estimate of population recordings of neural responses from each structure. And so this is something that we focus on in the course of the paper is population analyses of these data. But I think that it's something that's important to mention straight up, is that the analyses that we present in the paper don't actually rely upon the fact that these neurons are simultaneously recorded. So all of the analyses that we do are on so-called pseudo-populations, which are done by averaging the unit's firing rates by different conditions and then stacking those populations across different recording sessions. And so if you wanted to try these kinds of analysis approaches on other types of data, it would be perfectly okay to start with single neurons recorded on different recording sessions. So it doesn't rely upon the fact that they're simultaneously recorded. On the other hand, the fact that they are simultaneously recorded might mean that subsequent analyses done with the data would capitalise upon that simultaneous recording. And what I actually want to do is to kind of spoil the story for you by telling you our interpretation of the story right at the start. So you only really have to listen to this talk for another 30 seconds to kind of hear our take on the paper. So what I'm going to show you is a task that looks like one of those classic 2 alternative forced choices. And what we find is that the representations are very distinct between the 3 prefrontal subregions, and they're also very heavily shaped by the subject's current attention. But perhaps the surprising thing is that we don't think that the subjects are solving this task just by direct value comparison of the 2 options in that kind of classic neuroeconomic framework. Instead, we think there are features both of the behavioural data and also the neural data that suggest to us that the subject might be instead making a kind of sequential decision about whether to accept or reject the option that they currently think is best out of the 2 alternatives. And so I'll try and highlight those pieces of evidence as I go through the talk that have led us to take this kind of interpretation of our data. Okay, so that's the story, and here's how we're going to get there. So the task works like this. Subjects have to choose between 2 options, one positioned on the left and one positioned on the right, which are framed by those green rectangles. And each option consists of 2 pictures. And those pictures provide the subject with information. So one picture tells the subject about the amount of juice that they'll receive if they choose that option, but the other picture tells the subject about the probability with which they'll receive that juice. And the pictures are going to be randomly sampled from this picture set, which is shown on the left. So the subject has already learnt the meaning of these pictures very well before the experiment has even started, so there's no learning which is taking place during the course of the experiment. So in this example, he's choosing between a 70% chance of getting 1 drop of juice on the left option versus a 10% chance of getting 5 drops of juice if he chooses the right option. So if you're new to this field, maybe this would be a good time for you to pause the video and maybe just think which of those 2 options do you think would actually be the best option for the monkey to choose if he was kind of optimal at solving the task. Okay, so if you've had a bit of a think about that, well, the optimal solution for solving this task is to compute what's known as the expected value of the option, which is basically to multiply the probability of reward with the magnitude of the reward which is on offer. And so you might think in this case that means that the best thing to do is to take let me try and work it out. The left option, because 0.7 times 1 is greater than 0.1 times 5. But actually, there's a slight twist to that, which is it also partly depends upon how much you care about different magnitudes of reward, so how much you scale the magnitudes of reward. And so what we did when we set up the experiment was we tried to make it so that subjects would basically care equally about reward probability as much as they would about reward magnitude in making their decisions. And that's what you see in the behavioural data. So in this case, the subjects would kind of pay equal attention to reward magnitude and reward probability, and that kind of you'd have to factor that into the expected value equation if you were wanting to work out which option you would want to choose. So if you want to read more about this, then there are countless books on this, but maybe one of the best textbooks to start looking into studying expected value would be the neuroeconomics textbook, which is edited, I think, by Paul Glimcher and Ernst Fehr. Okay, so back to the task. So the trick in our task is the subject isn't going to see all of those pieces of information at once. So instead, we're going to reveal the information sequentially to the subject, a little bit like the shopper in the supermarket was attending sequentially. So the first thing that appears on screen is this fixation point. And once the monkey's acquired that fixation, the 4 picture locations are going to appear, but 3 of them are going to be covered with a grey square, and 1 randomly selected location is highlighted with a blue square. And that blue square instructs the subject to make a saccade to that picture location in order to reveal the picture that's hidden beneath. And he then has to fixate that first cue for about 300 milliseconds. And after that, the picture gets covered up. And then one of 2 things can happen. So in half of the trials, another blue square appears on the same option as the first cue. And the subject then saccades to that square, revealing the picture beneath. And at this point, he knows all of the information, both the reward probability and the magnitude, about that particular option. So, so we're going to refer to these in the paper as option trials. Okay, so that's half of the trials, and then on the other half of the trials, the blue square is going to appear on the other side of the screen. And if this happens, the subject has now learnt all of the information about one attribute, in this case, reward magnitude, but he doesn't know anything about the other attribute, so we'll refer to these as attribute trials. And then, once those first 2 pictures have been viewed by the subject, the 2 remaining pictures become highlighted, And the subject now essentially becomes free to do as he chooses. So he can either go ahead and gather the remaining pieces of information by saccading towards them, or at any time he can terminate the trial by moving the joystick that he's got in his hand in order to choose the appropriate option just based upon the information that's been gathered thus far. Once he's made the choice, then all 4 pictures get revealed to the subject, and then after a short delay, he'll get juice reward probabilistically delivered with a reward magnitude of the chosen option. Okay, so that's how the task works. I'm not going to spend a huge amount of time on subjects' behaviour because I want to get onto the neural data, but I'll just quickly mention a couple of things. So first of all, the subjects know the values of the cues very well, and they use this to their advantage. As shown in the supplementary information, they seem to use both probability and magnitude approximately equally to guide their decisions, and they end up selecting the option with the higher expected value on about 80% of the trials. The exact figure depends quite how you calculate expected value, so you can look in the paper for the final figure, but it's of the order of about 80% of trials. On the other hand, subjects don't tend to gather all of the information before they make their choice. And in fact, on only about 15% of all the trials do they invest the effort to look at all 4 cues before committing to making a choice. And on the remainder of the trials, they gather partial information before they make their decision about which action to make. So that then kind of raises the first behavioural question, which is how do they decide how they're going to gather that information as they make their decision? So this is what we look at in figure 2 in the paper. And we can ask this question in a couple of different ways. So the first thing that we can do is look at those attribute trials, which is where they've seen 1 cue on each of the 2 options. And the question that we can ask here is, where does the subject look next? So is the subject going to look back at the first cue, the first side that they attended to? In other words, are they going to saccade diagonally? Or are they going to saccade vertically and look at the other cue on the second side that they attended? And so to look at this, what I'm plotting here is the probability that the subject saccades diagonally back to option 1 as a function of the value of the first cue on the y-axis and the value of the second cue on the x-axis. And I've transformed those into ranks. So 1 is the worst ranked picture and 5 is the best ranked picture. And so what you can see is that when option 1 is high in value, but option 2 is low in value, then the subject tends to make a saccade diagonally back towards option 1. But on the other hand, when option 2 is high in value and option 1 is low in value, the subject tends to saccade vertically to gather more information about option 2. In other words, subjects tend to gather more information about the option that they kind of currently want to choose. And this is a behaviour that might kind of remind you a little bit of, confirmation bias style effects in psychology. But it's also a behaviour that we've seen in a human version of this task, which we collected in this large data set via a smartphone app, which we published a couple of years ago in PLOS Biology. And that paper also has some important controls built in for how informative the different locations might be in terms of revealing information about which choice you should make. But nevertheless, the humans still show this behaviour of gathering information about the option that they think that they might choose at the end of the trial. Okay, so that behaviour might seem quite intuitive. It might seem a little bit unsurprising. But now let's look at another behaviour. So this is what's shown in the right-hand side of figure 2. This is now on the option trials, where the subject has seen all of the information about one of the options. So they've seen two cues on one side. And what we're going to plot here is not where the subject looks, but instead how much more information, or how much information in total, does he gather before committing to making a final choice. And so that's what we're plotting here, again as a function of Q1 value and Q2 value. So this is the total number of cues viewed before the subject commits to making a final decision. Okay, so maybe this might again be a good opportunity just to pause the video and think, what do you think this figure should look like if you were optimal in terms of when you decided to committing to making a final choice? Okay, so if you've had a bit of time to think about that, now let's look at what the data tell us. So down here you can see a behaviour which seems to make sense. So when both cues are good, subjects don't need to gather more information. They already know that the side that they've looked at is the best possible option. It contains the best possible probability, the best possible magnitude, so they can go ahead and choose it. But the surprise happens up here in the top left corner. And that's where Q1 and Q2 both have the lowest value. So here, again, the subject has seen enough information to tell him which option he should choose, because he's seen that this option contains the worst possible probability and the worst magnitude. And so he could just go ahead and move the joystick and select the other option. But surprisingly, this is actually the situation in which subjects tend to sample the most information. So again, it seems like subjects are wanting to gather information about the option which they're eventually going to choose, even in situations in which we know in which he knows which option should be chosen. By the way, I apologise for always referring to our monkeys as he. It's just because both of the monkeys in this experiment happen to be male. We also see a related behaviour, albeit in a slightly milder form, in our recently published human dataset. So our current interpretation of these results, in both the humans and in the macaques, is that subjects have an idea about which option they currently want to choose, and then they seek out information about whether or not to accept or reject that item. And although, in the case of two alternative forced choice, that can produce some strange biases in behaviour, as we see here, if we think outside of the domain of two alternative forced choice, if we think about naturalistic decisions that we might face in real-world choices, this actually makes sense as a strategy for how to solve those kinds of choices. Because in general, if you see evidence against one alternative in a decision, it doesn't tell you which other alternative you should instead choose. So if you were to think about decisions with more than two options, or decisions where options weren't mutually exclusive, that framework of deciding whether to accept or reject a current best alternative is actually a very naturalistic way of making those decisions. And we also think that it might be the kind of way in which primates make those similar kinds of decisions when they're out foraging for food. And so two alternative forced choice kind of places subjects in this slightly weird scenario where actually seeing some evidence against one alternative tells you that the other alternative is the better one to choose. But our subjects might have such a strong prior of using accept versus reject strategies in their information sampling and choice that it gives rise to the kinds of behaviours that we see here and in the human data set. And we also use this to interpret some of the neural data that you see in the ACC a little bit later in the talk. Okay, so now I'm going to switch gears slightly and go to the neural data. So as I mentioned, we were recording from all of these three subregions, ACC, OFC, and DLPFC, and we had a reasonable sample of populations of single units from each of the 3 different subregions. And the analyses that I'm going to focus on today are those in figures 4 through 6, which are some ways in which we found were extremely fruitful in terms of understanding the structure of population responses in each of those different subregions. So the first analysis that I'm going to focus on is the one shown in figure 4, which is where we perform what's called a representational similarity analysis on the data. And this is a way that allows us to explore how information is coded across the neural population without adopting any real strong kind of prior assumptions on what that coding should look like, so how the representations might differ across the different regions. So RSA, representational similarity analysis, might be very familiar to some listeners, but I'll quickly walk you through it for those of you who haven't encountered it before. So the way it proceeds is like this. We can look at all of the trials where, say, the 0.7 probability Q, for instance, has appeared on the left-hand side of the screen. And let's do this for all of the neurons that we record in orbitofrontal cortex. So what we're plotting here is the z-scored firing rate for each neuron as they respond to this cue, so 100 to 500 milliseconds after the cue's been presented, along the x-axis. So each dot here corresponds to one neuron. And we can then move to a different set of trials where the 0.7 probability Q is now going to be presented on the right-hand side of the screen rather than the left, so completely independent trials. And that's what we'll plot on the y-axis. And what we can then do is we can measure, compute some measure of how similar those population codes are to one another. So in the paper, what we use is just simple Pearson's correlation. So that's what's shown by this slope here, is when we correlate the 2 responses together, what we find is that there's a positive relationship between those 2 population codes. In other words, the OFC population seems to respond similarly, or has some similarity to this stimulus, irrespective of whether it was on the left-hand side or on the right-hand side of the screen. If you want to get into RSA, it's worth noting that there are other ways of computing similarity. You can look at things like Mahalanobis distance or cross-Nobis distance or Euclidean distance between those different quantities. There are various reasons why some of those are slightly better or worse than other ones, but in our case, they produce broadly similar results. So the precise methodological details we've kind of left slightly to one side for this paper. But if you're interested in doing this for, say, fMRI data, then it becomes very important to think about exactly which measure you're going to use. Okay, so that was a little aside. We can then shift our focus to a different set of trials where, for instance, a different stimulus is going to be presented on the left-hand side of the screen. Let's say the cue that represents 1 drop of juice. And plotting this on the y-axis now seems to give us a slightly negative relationship with the population response for the 0.7 probability Q on the same side of the screen. Okay, so that's the basic idea. We're calculating these correlation coefficients between different conditions, and we can then go ahead and repeat that procedure for all of the different possible cues and all of their different combinations, and we can use that to build up a matrix of correlation coefficients that shows us how similar the population response is to different cues when they're presented in different locations. And that's what we see here. So each pixel here reflects the correlation coefficient between 2 different conditions in the orbitofrontal cortex, between the neural population response. And again, this is 100 milliseconds to 500 milliseconds after the stimulus was first presented. And what you can hopefully see is that there's already this kind of quite beautiful structure that seems to emerge in this matrix, which takes a little bit of time to get your head around. So just to orient you as to how the matrix is sorted, we've got the 5 probability and magnitude cues, and they're sorted from lowest value to highest value. So 1 is the lowest ranked value, 5 is the highest ranked value. And then we've again sorted them by when they're presented, whether they're presented on the left-hand side of the screen, which is on the top, or whether they're on the right-hand side of the screen, which is on the bottom. Okay, so again, I'm going to encourage you, if you're kind of new to this data, to just pause the video at this point and try and identify what you think are the kind of interesting things that kind of pop out visually from this matrix. Okay, so the first thing that you might notice is obviously this very bright diagonal. This is actually a saturated diagonal, so it's actually a diagonal of 1s. And this is just 1 by definition, because the correlation of something with itself is 1 by default. So the diagonal isn't actually that interesting, even though it's obviously visually quite salient. What you can do is, if you want to, you could split the data into 2 halves, and you could calculate the similarity between those 2 halves of the data, and then the diagonal might be slightly more interesting because it would tell you about the kind of noise in your data, the signal to noise, because it would tell you about what's the correlation between 2 identical conditions. But we haven't done that in this instance. Okay, so that might be the first thing which popped out, but then there are some more interesting things in terms of the task. So first of all, we see these off-diagonal elements here, and these indicate that there's representational similarity across the OFC population for the same stimulus identity, irrespective of whether that stimulus appears on the left-hand side or the right-hand side of the screen. Okay, and we also see this gradient of responses as kind of squares that repeat themselves across the entire matrix. So what these reflect is a value response for the currently attended cue, irrespective of its spatial location, and also irrespective of whether it's a probability cue or a magnitude cue. In other words, it seems like low-valued pictures are positively correlated with other low-value pictures. High-value pictures are correlated with other high-value pictures, but low and high value pictures seem to be anti-correlated with one another. It's also worth mentioning another kind of methodological detail, that whether or not that anti-correlation actually depends slightly upon whether we do that z-scoring or not. Negative correlation here should really be interpreted as being less correlated than other conditions, rather than there being negative correlations per se in the data. That's just something which is referred to in the RSA literature as kind of a cocktail blank effect. So if we hadn't done z-scoring, we'd see all of these correlations shift positively, but you'd see exactly the same pattern of correlations in the matrix. Okay, so methodological details aside, the 2 features that we've seen pop out in this RSA matrix are the attended stimulus identity, the off-diagonals, and the attended stimulus value. And that's in orbitofrontal cortex. But what about the other regions that we recorded from in the task? Well, if we repeat the same analysis for both the anterior cingulate cortex and the lateral prefrontal cortex, we see a very different set of responses. So first of all, neither of those 2 regions seems to have that strong stimulus identity code, the off-diagonal, that we saw in OFC. In ACC, we do have some evidence of something a little bit like the value code, but interestingly, it seems to take on kind of more of a block-like structure, almost as if it's kind of categorizing the cue into, is this a cue which I like? Is it one which is kind of above average, so a 4 and 5 valued cues? Or is it something which I don't like? Is it something which I might reject? the 1 and 2 valued cues. And so we'll come back to that a little bit later when we look at the ACC signals in more detail. And then lastly, we can see that the dorsolateral prefrontal cortex seems to carry some information about the currently attended spatial location. So basically the main thing which drives DLPFC is really whereabouts the subject is currently looking, whether they're looking at the left or the right-hand side of the screen. What's nice about doing this with electrophysiology data is we can also repeat this process through time, and we can see how these different representations emerge relative to one another as the subject deploys their attention to that cue. And we can also get a bit more formal about the different representations that I just mentioned, and the way in which we do this is to build templates that capture each of the different features that I just mentioned, and we regress those 4 templates at each point in time onto the RSA matrices. So those are the templates that you can see in the bottom left. I should also point out that we regress out the mean diagonal, and we also regress out the mean. We also regress out a constant. So those are two terms of regression model which aren't shown in this figure. But all of those 4 terms are included in a multiple regression. And what's going to be plotted here in the bottom right of this video, once I start playing it, is going to be the coefficient of partial determination, which is kind of a measure of variance explained by each of the different regressors having controlled for the variance explained by the other regressors. Okay, so let's start this video. So this is leading up to the time when the monkey makes their saccade. And what you can see is that the template that captures the deployment of spatial attention emerges really quite early. So this is the time when the saccade was being made, and you can see that this is really linked to the time when the eye makes the movement to the stimulus location rather than anything to do with stimulus processing. On the other hand, those other representations of stimulus identity and value and the accept-reject code in ACC, they all seem to emerge more or less in parallel with one another, and they kind of peak about 300 milliseconds or so after the stimulus is presented. So it doesn't really seem like any one of those different features seems to lead or lag any of the other features, although another feature, the left-right value, does seem to be slightly later. That's something which we refer to in the paper. I'll just play that video again one more time, just so that you can watch it now that you know what it's about to show. So again, this now is the time when the saccade is being made, and you see DLPFC really carries that spatial attention code very early on. And now, about 300 milliseconds or so, the codes kind of really emerge simultaneously across all of across both OFC and ACC. And at 300 milliseconds, the stimulus then flips back to being a grey square, and another stimulus location appears, and then after a couple of hundred milliseconds, the monkey will saccade to the second cue, which happens about now. Okay, so that's basically the first 300 milliseconds of the trial when the first cue has been attended. So in the remainder of the talk, I'm then going to go on to look at figures 5 and 6, which examine how these representations change as more cues are being attended by the animal as he gathers more information towards making a final choice. Okay, so let's consider this in orbitofrontal cortex on the attribute trials. And these are the trials, to remind you, where the subject is shifting his gaze from looking at one option to the other option. And so the first thing that we can ask is, how does value coding for each of the cues emerge across time as the animal shifts his gaze? So we do this simply by regressing the value of each cue onto the firing rates. And we plot the CPD, the coefficient of partial determination, or variance explained, as a function of time. So this is the mean plus or minus standard error across all of the neurons in OFC, time-locked to the presentation of Q1, Q2, Q3. And the response that you can see is very similar to what I showed you in the representational similarity analysis. In other words, the neurons respond to the value of a currently attended cue, and they peak about 300 milliseconds after the cue has been attended. But the other thing that you might notice is that when the subject shifts their gaze to the second stimulus, it doesn't return all the way back down to baseline. In other words, OFC still seems to have some memory for the value of a cue which was previously attended. Okay, so what we can then ask is whether the neurons which are responding to the presentation of the first cue, which are reflecting that value of a first attended cue, are also the same neurons at the second cue that respond to the second cue's value. And that's what's shown in figure 5b. And so the way that we do this is simply plot the regression coefficient for the value response at each time point. What you'll find is that they're extremely strongly correlated, with a correlation of about 0.9. In other words, this is a stable population code that stably reflects the value of the currently attended stimulus across Q1 and Q2. But what we can also see, as I mentioned, is that when the subject shifts their attention to the second cue, the value neurons don't return back down to baseline. I still have some memory of a previously attended cue. And so we can now ask the same question about whether these population codes for these two variables are related to one another. And we find that they are, but crucially, they're negatively correlated with one another. And that's what's shown in figure 5c. So this means that when I'm attending to the second cue, the neurons that code positively for the currently attended cue's value will be negatively correlated with the value of a previously attended cue, the first cue's value. And that was just at 2 time points, but in order to give a fuller picture of how that correlation changes across time, what we can do is we can repeat the same analysis across all different time points. So the warm colours here reflect positive correlations between different value codes, and cold colours correspond to negative correlations. And so B refers back to panel B here, and C refers back to panel C. And what's important is that if you then go and compare this with the other regions that we recorded, DLPFC and ACC, what we see is that although all of our regions have stable population responses that care to some degree about the value of the currently attended cue, only OFC has that really strong negative correlation reflecting the value of currently versus previously attended items. So it seems like OFC, but not ACC or DLPFC, is reflecting this attention-guided value comparison process. And then you can also see, if you look in more detail in the paper, how this can evolve across multiple cues in the option trials when you saccade to say Q1 and Q2 on one option, and then you saccade across to the third cue on the other option. But I'm not going to go into that in this talk. Okay, so that's the story for OFC. What about ACC? So ACC is a region that's had an extremely wide range of different hypotheses put forward over the years as to how best to explain its function. I would say that not all of them are mutually exclusive from one another. And what we found is that there seem to be multiple signals that exist in ACC in parallel with one another. And one of the ones that's most interesting, I think, is this idea of belief updating or belief confirmation in the ACC, which is what I'm going to explain to you now. So to remind you, at the time when the subject was attending to the first cue, the ACC seemed to have this really interesting structure that kind of categorized items into cues that you might ultimately want to accept versus those that you might ultimately end up rejecting. And this idea kind of fits in with some other findings that we've recently seen in cingulate cortex, particularly in tasks where you learn across trials. So what's found in learning tasks across multiple trials is that ACC seems to really show strong activity whenever you see something which tells you should change your behaviour, when you should update your beliefs about what course of action you should really be taking. And so that kind of made us wonder whether this signal that we were seeing at Q1 wasn't really reflecting value per se, but instead it was something kind of closer to confirming or disconfirming a hypothesis about whether to accept the option which is currently in the subject's foreground, the one which they first attend to. So this is again kind of speaking to that idea of decision making from an accept versus reject framework. Okay, so that's the kind of hypothesis, but what we sought to do was to try to test this idea in more detail as the subject progressed through the trial, as more cues were being revealed. And so the way that we set about doing this was to try to define what a signal which kind of looked like belief confirmation might look like. So we tried to define a regressor based upon the cues which were being attended, which would reflect how much the current cue confirmed the subject's belief. Okay, so let's try and define what that belief confirmation regressor might look like at the second cue on option trials. So if I've seen something good at the first cue on option trials, then seeing another good thing, a high-valued item, which you can see in green here, that confirms the belief that I already have. That tells me that yes, that's definitely an item I should choose. I've seen two good things. Whereas a low-valued item, which is in red, disconfirms it. So seeing something good followed by something bad kind of makes me a bit more uncertain about which option I should choose. But on the other hand, the opposite is true if I've seen a bad item at Q1. So now, seeing another bad item will confirm my current belief. In other words, if I see bad followed by bad, it confirms my belief. If I see good followed by good, it confirms my belief. And what's important is that means that this belief confirmation regressor that we've defined is now completely orthogonal to value, because on half of the trials it increases with the attended value, but on the other half of trials it will decrease with the attended value. We can also define a similar regressor on the attribute trials, where I shift my attention from one side to the other side of the screen. But here the sign of that belief confirmation regressor is going to be flipped. So in other words, now if I see a good first item, then actually seeing a bad second item confirms my belief about which option I should choose. And the converse is true if I saw a bad first item. So now if I see a bad first item and a good second item, that's going to confirm my belief. And we can play the same game at the third cue. So the same idea is true if we look at the third cue on option trials, but it's now defined based upon the integrated value of both Q1 and Q2. And then last of all, we can define it at Q3 on the attribute trials. And here, it actually depends upon a combination of Q1 value, Q2 value, and whereabouts the subject decided to look. So whether they decided to look at Q1 or Q2. Okay, so that may have all seemed somewhat convoluted, but what's important is that we've now got these four complementary ways of defining this idea of belief confirmation or belief updating. They're all defined at different points in the experiment. In fact, they're defined on completely different trials and at different points in time. And crucially, they're all orthogonal to value, so we can include them in the same regression model as we included the regression on value, and that's what we did. And so what was striking was that then if we go and estimate that regression model, and we go and look in the ACC for how much the neurons care about that belief confirmation regressor, then what we find is that their regression coefficients are all correlated positively with one another. In other words, across the ACC neural population, there seems to be this kind of stable population representation, or what we'd refer to as a population subspace, which reflects how much the currently presented evidence confirms or disconfirms your current hypothesis or belief. And the kind of correlation values for this are shown in figure 6, which shows a comparison between ACC and shows that it's not there in OFC or DLPFC, and you can also see the raw correlations in supplementary figure 10. And then finally, what we can then go and do is just take the firing rates from all of the neurons that we recorded in ACC, and we can project them into a subspace which is defined by those regression coefficients. So we take basically the data from half of the trials and fit the regression model, and then we take the left out data from the other half of the trials and we say, okay, how does the activity evolve in that space of, if I'm a neuron which cares positively about belief confirmation, I'm going to contribute positively to this subspace, and conversely, if I'm something which cares negatively about belief confirmation. So the way that we do it is kind of akin to taking a dot product between the regression coefficients and the firing rates. Full details in the methods, but basically you can think, if I go up on this y-axis here, it means that I have more of that belief confirmation code kicking about in my ACC at that point in time. So that's what we did, and then we saw how the activity within this subspace evolved on trials where the subject responds, where they move the joystick at different response latencies. And what's remarkable here is that the activity in this subspace always seems to ramp up prior to the time when the final action is being made by the subject. And you can actually see that it's kind of slightly suppressed in the trials where the subject takes a particularly long time before they commit to making a response. So we can slide this across so that it's response locked, and we'll see that it kind of peaks a couple of 100 milliseconds prior to the time when the choice is being made. So there seems to be that belief confirmation code confirming which choice I should make. But what's also interesting is that there's also neuronal activity in ACC, but selective for whether that joystick movement is going to be a rightward movement or a leftward movement. So we can split those trials into those where the subject chooses left or right, and we can then project our activity into a completely orthogonal subspace which reflects the emerging representation of which action is going to be chosen by the subject. And when we rotate our data into that subspace, we see that at the same time as the belief confirmation code is emerging, there's also this simultaneous representation of which action is chosen that's ramping up across time. So because these two subspaces are orthogonal in a supplementary movie, we get fancy and we plot them both in 3D to see how they emerge across time relative to one another. And then once we've learned how to make MATLAB movies in 3D, we also spin them around. And that has no scientific value whatsoever, but it's just there to act as eye candy at the end of a talk. Okay, so this is a summary of the parts of a paper which I've focused on today. We have recorded from 3 subregions of prefrontal cortex in this attention-guided decision-making task. We think that there's some evidence to support this idea that the way our subjects were solving this task was more in this kind of framework of a decision in terms of whether to accept or reject the currently attended option. And we also found very different roles for each of the 3 subregions that we recorded from. So dorsolateral prefrontal cortex seemed to reflect whereabouts the subject was currently deploying their spatial attention. OFC population responses seem to reflect this value comparison between the currently attended item versus items which were previously stored. And anterior cingulate cortex population responses had these kind of parallel subspaces, one of which showed this belief confirmation code ramping up prior to accepting a choice alternative, and another one reflecting which action the subject was going to choose. I just want to finish with a kind of final point, because I appreciate that a lot of readers, hopefully a lot of readers of this paper, will come from slightly different fields and they may work with very different types of data. And I'd like to emphasize the kind of potential of this approach of representational similarity analysis as one way of bridging between the kinds of data which we've been studying in monkeys and things that we might try and measure in the human brain. So in particular, what I showed you in this talk was, and in the paper, was how we use monkey single cell recordings to study the representational structure of our data. But we're often faced with this kind of correspondence problem in systems neuroscience, where we're often faced with very different types of data, such as fMRI or MEG or computational models, sometimes coming from different species. And so an idea that's really been espoused most by perhaps Nico Krieger-Skorter is that by mapping these different types of data into this kind of common space of representational similarity, we might be able to draw direct correspondences between these very different types of data. And this has already proven extremely successful in the ventral visual stream for researchers studying object recognition. So here are two representational similarity matrices, one derived from monkey single cells, in a similar way to the way that I was describing in the talk, and another one derived from human fMRI voxels during an identical task. So whilst the subjects were asked to just attend to different types of visual stimuli. This has also been applied to other types of data, such as human MEG data. So what I'm hoping is that in the near future, some of the representations that I've showed you today might also be identified directly in the human brain using similar approaches. And this is, along with several other studies, is a line of research that we're currently pursuing in my new lab at Oxford, where we are primarily running human imaging experiments, but continuing to collaborate with Steve Kennerley's lab and other labs to do electrophysiology experiments. And this is based in this fantastic new Wellcome Centre for Integrative Neuroimaging that's recently been founded. So we have this a fantastic setup here in Oxford with great collaborators. And please, if you're excited by this work and you're kind of on the lookout for potential postdocs, for instance, if you're a grad student who's on the lookout for that, or you're interested in getting in touch, please do email me, and I'd be delighted to hear from you. Okay, so with that, I'd just like to finish by thanking again the key collaborators on the project. So Nish Malalasekera, who's joint first author on the paper, who, as I mentioned at the start, really led the charge on the data collection. Steve Kennerley, whose lab in UCL, where this data was all conducted, this project was all conducted, and also Tim Behrens, Archie de Berker, Simon Farmer, and Bruno Miranda. And I'd also like to thank my funding agencies, Wellcome Trust, Brain and Behaviour Foundation, and NIHR. And thank you for your attention. Please do get in touch if you would like to ask me any questions about the paper, and if you want to do any further analyses. But with that, I will sign off. Thank you.